Here in the auditorium in Rio, and welcome to all of you joining us online for the final day of our third BBNJ symposium. No, Andre, it's not a good thing. We're running out of time for all our fantastic discussions and contributions. Should I give everyone just a couple of minutes? Or actually, I think my announcement has done the trick, and there's actually quite a good audience. So I think we will formally begin day 3 of our symposium. So I just wanted to say a couple of things before I introduce our keynote. I'd love to hear how it's going for all of you. You know, there seems to be so much discussion outside of the sessions. It's really encouraging to see. But, you know, as part of the hosting team from Mara Consultants, it would be really great to hear from you what you're getting out of this meeting so that we can look ahead to future meetings. I think I'm going to go ahead and introduce our keynote now. Yeah, and say things later on. All right. So, everybody, I am... okay, we're just testing through some panels. We're just having a checking some of our panels. What's coming up later? This is a great advert for panel 9. That's actually our final panel of the day before closing. Fabio, if you can, we would like the panel, the slide for our keynote this morning. While we get the slide up, let me introduce our keynote speaker to you. We're very delighted to welcome Margaret Leinen to the stage. Margaret is Director Emeritus from Scripps Institute of Oceanography and Vice Chancellor Emeritus for Maritime Science at the University California, San Diego. Now, Margaret, I know many of you will know Margaret very well because she has had a really illustrious career in scientific leadership. She's previously served as Assistant Director for Geosciences of the US National Science Foundation and as the President of the American Geophysical Union and the Oceanography Society. She chairs the Ocean Biomolecular Observing Network, a program of the UN Decade of Ocean Science for Sustainable Development, and she has served as co-chair of the Decade Advisory Board. We're going to try and leave time for maybe one or two questions from the audience at the end of Margaret's talk, so please get those questions ready. And it now gives me great pleasure to hand over to Margaret Leinen.
Thanks.
We'll just wait for your slide.
Thanks, Hermione. There we go. So the program says that I'm going to talk about science and implementation. And I've actually adjusted this talk a little on the basis of things that we've been talking about the last 2 days. And we've heard a lot about the focus of BBNJ on the best available science. And so I want to talk a little bit about implementation with the best available science. And just to remind you, almost all of the mechanisms in the framework of BBNJ rely on the scientific data. We focus on biodiversity baselines, species inventories, the identification of priority conservation areas, assume we know something about the biodiversity. So I'm going to talk a little bit about that. How much best available science do we have to work with for BBNJ? In large regions of areas beyond national jurisdiction, the empirical occurrence data are extremely sparse. So where are the data? We tend to think of it in terms of these databases, the OBIS database, in the case of, for example, the Clarion-Clipperton Fracture Zone, the ISA Deep Data base, fish base, sea life base, which are really more traits and ecology rather than occurrence, and aqua maps, which you heard from Jesse Cleary about Tuesday for predicted distributions. And I apologize for how difficult this is to see, it actually looks a lot better on the computer, and I tried redoing it with a different color scheme just before this, after I saw this, but it looked even worse. I hope you can make out the white continents, and then there's a faint gray color to the background. And this is all the occurrences in areas beyond national jurisdiction in OBIS. All of the places where you see that gray background, there's no data. Each little point is a 1 by 1 degree square, and the colors are the number of observations in the square, so it's a heat map. And the yellow colors are less than 10 observations. So this is, Alex talked about this yesterday morning, that there are huge areas, essentially most of the Pacific, most South Atlantic, the Southern Indian Ocean have almost no data in the OBIS database. So there are 160 million records. There are about 12 million in areas beyond national jurisdiction. Of those grid cells, there's 64,000 grid cells, 1 by 1 degree. 65% of them are in areas beyond national jurisdiction. 70% of those have no data, and 90% have fewer than 10 observations. So the idea of really easily getting a lot of scientific data on which to base that best available science is a real challenge. This is another way of looking at it. On the left, that is the data sets in OBIS, and there are about 2,700 data sets. The blue are all the areas within national jurisdiction. The yellow is the areas beyond national jurisdiction. Only 3% of the data sets are there. And then the gray is data sets that straddle both in and out of national jurisdiction. On the right-hand side is the species data, and even less there. Very small amount of the species data and the data sets are in areas beyond national jurisdiction. So, and then each, if you start looking at individual groups of organisms, you see other kinds of asymmetries. So this is a diagram that shows all of the taxonomically resolved phytoplankton samples in OBIS. And again, you can see here, phytoplankton, one of the most abundant groups in the ocean. And you see that aside from the North Atlantic, and that's almost all continuous plankton recorder data, it's empty. There's not a lot there to be able to look at the base of the food chain that the larger organisms depend on. This is another interesting element is how frequently areas are studied. So the top is a map of sites that have been monitored for at least 5 years. So you can see here as well that virtually all of them are coastal. They're in areas within national jurisdictions, only a few sites outside. And the lower diagram is the stations added to OBIS. So we've done a lot more. We've been adding a lot more, especially in the last 20 years or so. But the proportional change in the number of stations surveyed has been going down. That's the blue curve that then turns into red because of the lack of continuous monitoring. Another example, this is the Clarion-Clipperton fracture zone area, so the deep sea mining area. There's Kiribati here on the left, Hawaii up at the upper left, Mexico EEZ on the right. And you can see the outlines of all of the lease areas. The yellow dots are OBIS data. There are exactly 15 places where there's OBIS data. The blue dots are GBIF data. There are 11 of those. The green are ISA's data, deep data. There are 25 of those. And all of those turquoise dots are literature references. So the author, Muriel Rabon, actually did a literature search on available data. This area that covers the whole deep sea mining area is 2/3 the size of Brazil. It's 108 data points. Imagine trying to characterize the ecosystems of Brazil with 108 data points. That's the challenge that we have. And some of there's some good news. There are many more species that are being identified, a lot of work that's going on. It's just that the amount that is easily recoverable in databases is not much. And then on the right-hand side, those are taxonomic groups like the annelids and the arthropods, mollusks. The orange are unrecognized species. The blue are named species. So even when we recognize that we've got something, we often haven't described it. And then one other thing I wanted to comment on, again, Jesse on Tuesday referred to Aquamaps, which is, Aquamaps is a tool for being able to predict biodiversity. And what it does is it looks at the habitat, temperature, salinity, depth, oxygen, all of the characteristics, and then it maps species that live within those parameters onto the map and tells you what the predicted biodiversity is. So the red colors are higher biodiversity, yellow, lower biodiversity. And as Jesse mentioned, this is also something that can be used to look at changes in the future. But when we talk about this and then look at the data that's available, you understand how tentative these maps are. So some of the concerns about this. The STB is going to need to think very carefully about the standards that they're going to have for analysis of proposals and EIAs, because they could really box themselves in by setting very high standards when there's not a lot there. It'll be difficult for them to assess proposals without substantial effort to look beyond OBIS and the other databases. Most of the biological data is not in OBIS. And I was talking to a scientist yesterday. We were talking a little about this situation. And they said to me, very little of my data is in OBIS because it's so hard to get data into OBIS. It requires a lot of work. And it's not just that you have to go through it all and you have to enter it. There are many processes that go into getting the data into OBIS, getting through the Darwin core requirements, the WORMS database, and so forth. So it's very difficult. And given the paucity of data, there may be substantial pushback, or Angelique yesterday used the term relitigation from STB to COP. So think about all of the stakeholders out there that aren't really enthusiastic about setting aside areas. And the lack of data could be an important tool for them to push back. And given the lack of monitoring in areas beyond national jurisdiction, the STB is going to have to think very carefully about setting standards for evaluation after the fact. So these are some realities that I think that we have to accept. We have to think carefully about them, and as time goes on, I think that things are going to improve. But at the beginning, it's going to take a lot of work. And who will be responsible for that? However many people are going to be on STB are not going to be in a position to be able to do that kind of literature work. Are they going to require it of the community? Is there going to be, are contractors going to do it? I think that we need to think about the reality that we may need a function somewhat like IPBES or IPCC for BBNJ to be able to do this hard work. What can we do? Get more of the data into databases, but as I said, that's a real challenge. And I think that one of the things that is changing is the acknowledgement that data need to go into databases, but we're still also faced with many databases that don't interoperate. For example, in the US, the National Science Foundation requires that biological and chemical data go into a special NSF database, but that database is not interoperable with OBIS. So we have to get more agreement on this, or we have to build the tools to allow these databases to be interoperable. And that's separate from the clearinghouse mechanism. A tool is not the mechanism. So some of the enabling developments that I think we're going to see improve the situation are certainly environmental DNA and other biomolecular observation. Sejan talked about this yesterday. Seems like a week ago. Autonomous platforms, and they definitely are being developed. Ship of opportunity sampling, and then really getting these databases together and standardizing metadata. So I'd like to take 2 minutes to tell you about a development that I think is poised to really change this situation. And that is the use of environmental DNA and other biomolecular techniques. So I chair one of the UN Decade programs called the Ocean Biomolecular Observing Network. It is not the observations themselves, it is the tools that people need to be able to do that. It's the network of all of those programs. So we have 27 programs that are now are monitoring almost 200 time series sites. Most of them are in coastal waters, not open ocean, but it's starting to really develop. So this depends on the fact that every organism in the ocean sheds nucleic acid into the water. It either excretes it, or like the marine mammals on the lower right have a slime coating or mucus coating that contains DNA that goes into the water, or the organisms release nucleic acids when they die. And so we're able to then sample the water instead of trying to catch, photograph, or otherwise capture the organisms. And we have several tools within the biomolecular community to be able to identify organisms. So this is really opening up the ability to much more easily understand the biology. And the cost of doing this is now low enough that that many, many more people can access these techniques. So the network is building from members of the community a multi-omics biodiversity observing system. We have had 2 workshops on developing the autonomous technology that Sejin called for yesterday. When we started 5 years ago, there were 3 autonomous samplers. There are now 17 with a range of capabilities. So that's how quickly this field is evolving. We are working to enhance the data systems, building tools to be able to get the data into OBIS a whole lot more easily, and by adding other biomolecular components. We've been partnering with marine conservation and management specialists, groups like this. OBAN certainly wants to partner with the BBNJ community to be able to assist in getting data available, and then develop and transfer capacity through training programs supported by development aid agencies, philanthropy, and so forth. And we're doing this both in person and online. So what else can we do? I think that we have a historic opportunity to align governance with other observing opportunities. And I'll just highlight 2. There's a developing initiative led by France called Neptune Mission, which is focused on high seas, full depth, and biological observation. That sounds like BBNJ to me. And there are many nations that have already agreed to participate in this. And it has several themes. For example, seamounts is one of the themes, polar areas is another, areas of high productivity, like the thermal dome that we heard about yesterday afternoon, equatorial Pacific, and so forth. InSync Antarctica is an international initiative to deploy multiple ships in the circumantarctic region between 2027 and 2029. And it already has an eDNA working group to really speed biology. So that's going to mean that BBNJ needs to think about partnering with CAMLR as well. It's one of the other important treaties that I really didn't hear discussed, but the Antarctic waters are some of the most productive in the world, and we can't ignore those either. The lines on this map represent the transects of what has been a physical and chemical set of observations, really the backbone of GOOSE for physics and chemistry, called GOSHIP. And those lines are occupied every 5 to 10 years to provide snapshots to look at change in the ocean. And GOSHIP has now added eDNA to its array of observations, and now says that BioGOSHIP wants to become an international collaboration to measure, understand, and predict the distribution of and biodiversity of pelagic communities. That was announced at Glasgow the week before last. So although that's not going to give us the fine-grained information that we need for BBNJ areas, it is going to give us a much better sense of the change in the biology of the ocean. And here's a list of the various measurements that are included. Some from underway measurements, some from CTD, some from labs. So it's not just eDNA, it's a wide variety of biological measurements. So this is my view of what we're going to have in the future, and that's a BBNJ observed by new observational tools and using a network committed to standards and open data. Thank you.
Thank you so much, Margaret, for raising some really important issues, so much in that talk. We definitely have time for some questions after that brilliant keynote. Does anyone here have a question for Margaret? Okay, Murray, could we get a microphone down here, please? Actually, I'll just give you mine, Murray, and then we'll take a question over there.
Thank you so much. Wonderful talk. eDNA is fabulous, a technique and approach, but as you noted in your talk, so many species haven't been identified or sequences. Just maybe to reflect a little bit upon that basic work that we need to do to make sure those sequences are in the databases for that deep and open ocean eDNA to hit and match.
Right. I think that the real focus has been to concentrate on the demand side. So one of the first primers that was developed was MyFish for fish, because everybody wants that data. So I think that it's really a conversation. Which are what's the greatest need? And I know for biodiversity, you want it all, but that's what the conversation is about. So I hope that, although OBAN is a decade program, there are no plans to stop at the end of the decade. We really see it as the framework in much the same way that that GOOSE has been a framework for physical and chemical observation, that OBAN can play that role for biological. And obviously it's going to be closely associated with GOOSE.
They've got a microphone over there. Okay, yeah, we have one more question here and then we will come, actually Bhupe? Okay, Bhupe, then Andre.
Thank you. Good morning, Margaret. So Bhupe from Basecamp, UK. Thank you so much for a wonderful presentation. And as our partners, our collaborative partners, it was really nice to hear on some of the updates, on some of the work that is going on. My question is, with similar type of collaborations that are currently the BBNJ is encouraging cooperation between private sector, public sector, academic institutions. Yesterday we heard from Brazil about their vision on what they wish to do. What would you say is the future for realizing equitable partnerships between these various different stakeholders and actors? What would be, you've been working in this space for so long, what words of wisdom would you be leaving us in encouraging equitable partnerships? Thank you.
I think the real key is the areas of common ground. And we had a great presentation on Tuesday. Casper's? Jasper, yeah. Marcel Jaspers. And he talked about the needs of the marine genetic resources community. And I saw several overlaps there with the interests of the OBAN community and how we can look for the areas that we're both interested in developing. And I think those are the places where we build the partnership and then let it flourish. Just in the same way that we do as scientists, I'm interested in one area and another scientist is as well. And so we start talking about our common interests and the partnerships flourish. So I'd really say that that's the key.
Margaret, that was super clear. Thank you very much for showing the gaps in knowledge that we have in the BBNJ, in the ABNJ, but most of all coming from the perspective of my passion, ocean science diplomacy. And when you think about the power that scientists have to sell to diplomats like the silver bullet solution for that problem, it becomes a kind of worrying that we are not delivering the right message, which would be that we need to strengthen our capacities in terms of observing those big areas, and that the time that this will consume is much longer than the time that diplomacy requires to make decisions, because we're going to probably have the first COP this year. So what would you say would be the most balanced message that we could deliver this as a BBNJ community back to the decision makers who are always changing into the process? How, as scientists, from your background, how do you think that we should frame this in a way that they can understand and we can talk to each other in a better way?
I always thought diplomats worked pretty slowly. I thought we might have more time than we think. I think that, we have to be realistic about what's there. And now, for those of you that were in the wonderful session on the thermal dome yesterday, obviously there was a lot of biological data that group had developed. They're interested. They exploited the fisheries data. They exploited years of research there. But it took them a long time. That was a major science effort. And so I think that we have to be realistic with the diplomats to say, we will get there. And if I start identifying the areas that you think you're prepared to work on, it will drive scientists to those areas. And especially if BBNJ is saying, we need this data, that'll be easier for us to make the argument to get it. And then, as I said, we're rushing as quickly as we can into more autonomous tools and more tools like eDNA. Some really terrific work. A fabulous study that's going on comparing eDNA for benthic organisms to photography and video, and looking at the positives and the negatives there to understand how to use these tools in that environment as well.
Mark, we're going to take two more quick questions. Jimmy Say, then Larry. I'm sorry, then we will be moving on to panel 7. So panel 7 panelists, if you could move down to the front.
Thank you very much, and thank you so very much, Margaret, for the excellent talk. It's very inspiring. I have to chair a session by the end of the day today, and that's on marine genetic resources, so you led the way for that. Personally, I'm much into the data infrastructure business of INPO. I'm the Director of Infrastructure and Operations, and my latest lover is eDNA. So we've been working, studying, partnering with other institutions so that we can evolve in that regard. My question is regarding infrastructure. I know it's not cheap. We may need lots of computing time to do the sequencing. And alluding to part 5 of BBNJ, do you have any idea on how could we pursue not just benefiting from the information from afar, but being capable as a developing state to perform those activities? Thank you.
Yeah, I think the real key, or what we're seeing is developing a few sites that have super sequencing capability, really large scale, really fast. And that drives down the cost, and it means that you don't have to have every university have that capability. And one of the things that has happened is that some philanthropies are investing in the core facilities so that they can speed up the spread of the use. So I would say, concentrate on getting really substantial capabilities somewhere, and then just drive all of the analyses to that center.
Great, thank you. Final question, Larry?
Thanks, Margaret, and thanks for an amazing keynote. A very quick, boring lawyer's question, specifically on ships of opportunity. And how do we make sure that the scientists using those understand that the BBNJ obligations apply to that work as well? Because it's a bit of a headache, to be honest, for the existing MSR procedures to make sure that's done. Thanks.
Yeah, I'm not talking as much about citizen scientists, so somebody has a yacht and I'm going to go out on the yacht and maybe I'll take a sample or two. It's more, for example, shipping. We've had agreements from some of the major shipping companies to put an underway sampler on. And so if you have that, you do your QA, QC at the beginning, at the end, and have that capable of operating on its own. That's the kind of ship of opportunity stuff that I'm talking about. And in those cases, the scientists who are responsible for that are also very familiar with what the issues will be, the legal issues for BBNJ.
Thank you so much. Margaret, I think we could have taken 10 more questions, but thank you for a brilliant keynote. Thank you. Okay, well, I'm very pleased to ask the panelists to come up to the stage. Just as they do that, a note about Slido. We're going to be using Slido throughout the rest of the day. That's available to all of you online to interact with our speakers throughout the day. And also, feel free to use it if you're here with us in Rio. We're not able to get through all the questions, but Slido, the questions will be banked, and it actually provides a really useful resource for the organisers and for the panellists to see what questions are out there. So please do populate that Slido for a bigger purpose. Okay. Welcome, panel 7. I'm going to hand straight over to the chair, Julia Schutz-Vega, a researcher at the Nova School of Law. And panel 7 is about lessons learned from relevant institution, frameworks, and bodies, the IFBs. Right, Julia.
Okay, this one's yours. You have like 50 minutes.